All right. Hi, everyone. I'm Allie Metzger. I am a senior at Montrose High School. And this summer I was at the Rocky Mountain Biological Lab studying pollination ecology in the Brocy Lab with my amazing mentor, PJ Jones. Ooh, come on. Oh, okay. So what is pollination ecology? Well, it's the study of the relationships and interactions between flowering plants and their pollinators. So some key things you kind of need to know. Plant-pollinator interaction. So the relationship between plants and pollinators is a mutualistic one, which means both sides benefit from it. Plants offer food to pollinators in the form of nectar, and in turn, the pollinators will pollinate those flowers and help them reproduce. This forms what we call a plant-pollinator network, so you can see my lovely professional picture over there. It's basically links between pollinators and the kind of plants that they visit, so it's kind of just the structure of the ecosystem. And then finally, floral characteristics, which are just ways that flowers may have adapted to attract certain pollinators to them, so keep that in mind because that's kind of important for the next slide. Yes, I worked with my amazing mentor, PJ Jones, who is a Harvard graduate and is now a PhD student at the University of Washington, with her research question of, does the nutrients of pollen of different flower species affect why pollinators visit? So thinking back to the floral characteristics, have flowers adapted to have a certain ratio of nutrients in their pollen that attract certain pollinators to them? So this basically consisted of me using a makeup brush and a microcentrifuge tube and bending down really, really close to flowers and using that brush to brush pollen off the anthers of the flower, which is where the pollen is produced. We collected pollen from many different flower species and across many different sites. And this is kind of just to show the variation of flowers that we collected from. There was a lot. Once we collected the pollen out in the field, we would then have to bring it back to the lab to clean it. So first we would use the vortex and centrifuge machines. So the vortex machine spins the heavy stuff, so anthers and bugs that we sometimes get in the tubes out in the field. It spins that heavier stuff to the bottom and leaves the pollen up on top on the rims of the tube, so then we can dump out the heavier stuff without losing any of the pollen. And then the centrifuge machine spins all that pollen caught on the side of the tubes down into the bottom of the tube in a nice, neat little pile. If that was not enough, sometimes we would have to take a makeup brush out some of the pollen from the tube, pick through it with tweezers on the cutting mat, and then we would use a paint scraper to scrape it back up and put it in a clean tube. Other research, so the Brocy Lab was also doing some other research that I was grateful enough to be a part of. So at all the different sites that we visited, there were plots of land that were set up. And so they were kind of looking at if they change something in the plant pollinator network, that structure, how do the pollinators react, what happens? So basically in those plots of land, we'd first count the flowers and see, okay, what kind of flowers can pollinators visit? And then depending on if it was a control day or a manipulation day, on manipulation days, we would spray a certain species of flower with quinine, which basically makes it taste bad to insects and is making it unfavorable in the environment. If it was a control day, we wouldn't do that, and then we would basically observe little plots of land for a total of 3 minutes, and any time that we saw a pollinator interact with a flower, we would stop the timer, catch it, and we'd unfortunately have to kill it and ID it, unless it was a bee or butterfly. And then we would bring those vials with the dead insects back to the lab for insect processing, which was just moving the insects to a new container for the people there to bring back to college to do like the more research on it, I guess. Or we do pollen processing, which is collecting pollen off of the insects, so we could look at what kind of flowers that insect was visiting. Outside of the internship, in terms of housing, I was lucky enough to stay in a condominium with my amazing roommate, Celine, which was very, very close to Rumble. It was very pretty. I don't have a picture of the actual place, but this was basically my view out of the balcony there, so it was really very pretty. And then in terms of commute, so I was very close to Rumble and Snodgrass, but from Snodgrass to Rumble, it's kind of a long dirt road section. So thankfully, my mentor was kind enough to pick me up in the U-car there, which we're posing in front of. And she took me down that dirt road. So I only had a 5-minute bike ride from where I was staying to Snodgrass and back every day. And also there were free shuttles in Crested Butte. So anytime I needed to, I guess I was staying in Mount Crested Butte. So anytime I needed to get down to Crested Butte to do grocery shopping, I had shuttles. So that was really great. These are just some of the things I did outside of the internship. The Brosi Lab and the girls there were super welcoming, so I went hiking with some of them. They invited me out to dinner. We did a potluck. There were a farmer's market. I was invited to watch a no talent show. And yes, that is real, and and it is exactly what you think it is. It was very fun. And then something that surprised me was just the sheer variation in pollen between different flowers. So I was staring at flowers like basically for like multiple hours every week. So you see a lot of things. So we have Heraculum, which is the white flower, Helianthella, which is that yellow one, and Delphinium, that's the purple one. Heraculum has green pollen, Helianthella has bright yellow, and Delphinium has white. So it was just really interesting. I didn't know that there were different colors of pollen. And my advice for future interns is to take advantage of your mentor's expertise. I was there with PJ to study pollen, but she knew a lot about bumblebees, and so anytime I'd see one, I'd be like, oh, PJ, which one's this? So it was very cool. Don't be afraid to not get things right the first time. Scientific names are hard, and I had to know a lot of them for those flower counts. They're going to be patient with you. They don't expect you to know all those things. And then don't turn down new opportunities, like not only with work and research, but also if your lab mates invite you out to do something fun, take that opportunity. You're there to have a well-rounded experience. And then finally, special thanks to all of these amazing people.